I'm going to give you guys more kudos too, because I'm having trouble with my physiology class. They've seemed to have gotten really dependent on the recordings, and it's been affecting attendance. But that doesn't seem to be the case with you guys, so good job, because the next step with them is that I'm not going to put them up there anymore. So, because their grades also dropped in the class that's not attending, their grades dropped almost 10% from one test to the other. So there's not really quite a substitute for attending class from my perspective. I don't think it's a complete substitute, but anyway, you guys are still coming to class, so I'm going to keep putting these up here for you. Okay, so where we were is, remember we had just talked about the diencephalon, and I know it's been a while, and I know that term is not particularly meaningful unless you've learned it. So do you remember what the parts of the diencephalon are? Thalamus, hypothalamus, epithalamus, including the pineal gland, right? So this picture has both the diencephalon and everything inferior to the diencephalon, which is considered the brainstem. So it includes the thalamus. Here's the hypothalamus, because the mammillary bodies are part of the hypothalamus. And then the epithalamus is back here, right? Okay, so all of this that you're seeing that's kind of this light aqua color, that's still part of the diencephalon, and then we're going to move down to the stuff that's part of the brainstem, right? Which is the darker aqua color and everything inferior to that. Does that make sense? Okay, so if we move down to the brainstem, the first part of the brainstem is the part, basically we're going down on our evolutionary tree, right? We're going like to simpler functions. And everything close to the spinal cord are the oldest parts and also sort of the life-sustaining parts. So we're going to move more to that functionality now. And then remember, with our metaphor of the ice cream cone, we did the big scoop, right? And we did the chocolate coating, and now we're moving down to the cone itself. So this part is the cone that itself. If we're looking at a mid-sagittal view of the brain, where we would be talking about is, okay, so here's the pineal gland, right? And here's the thalamus and the hypothalamus. So the regions we're talking about are right here. One of the ways that I can find this region that I'm about to tell you about, which is called the mesencephalon, is to find the cerebral aqueduct, right? And it's the things that are just around the cerebral aqueduct. Does that make sense? Okay, so now let's look at what we're talking about. Okay, so the mesencephalon, or the midbrain, is really the superior portion of the brain stem. And it includes these guys right here, right, which are called the superior and inferior colliculi. You've got two superior ones and two inferior ones. Collectively, unfortunately, people can't agree what to call them, so I'll tell you both of the names. Collectively, because then there are four when you put them together, right? Two superior, two inferior. Collectively, they are called the corpora quadrigemina. And corpora means body, and quadrigemina means four twins, the four-twinned body. Do you see? It looks like there's four of them. In humans, they're all about the same size. If you looked at them in sheep, one of them's way bigger than the other. The top one's way bigger than the bottom one. So the superior, yeah, it's because sheep, these are for visual and auditory reflexes, and they have better visual reflexes than they do auditory reflexes. Like a lot more processing for visual reflexes than auditory reflexes. So, okay, so superior and inferior colliculi. So these are part of this region that's called the mesencephalon, but so is this part right here. So these parts right here, just lateral to the mammillary bodies, and they're actually lateral, and they're also a little bit more inferior. It's hard to tell depth when you're looking at this figure, but I'll show you another figure in just a second. So these guys right here are called the cerebral peduncles, and they really look like columns that go straight from the pons up to other structures, right? So they're columns on either side. They actually look like the push-up bra for the mammillary bodies too, right? If you can find the mammillary bodies and look at what's making them so perky, it's those things right there. So those are the cerebral peduncles. Now let's look at them in an actual specimen. Here they didn't draw them in, so that's one of the reasons I don't like that figure. But here you can see them, can't you? The cerebral peduncles just lateral to the mammillary bodies, right? So those are the cerebral peduncles. So we talked about the fact that where did my picture go? these superior and inferior colliculi do visual and auditory reflexes. So let's get that functionality before we get to the cerebral peduncle functionality. Top two are visual and bottom two are auditory, if I remember correctly. Somebody should check me on that because I may have them flipped. It's in your textbook, but I forgot which one does what. So if you don't mind, if you have a second to check it, because I don't want you to learn it. I mean, obviously, I'm not going to test you over which one does which if I can't remember, but I would like to remember, so maybe you can teach me. I think it's auditory and visual. Okay, so these guys, what do I mean by auditory and visual reflexes? So a reflex is different than a voluntary activity. With a voluntary activity, I'm engaging my cerebral cortex and making voluntary conscious decisions about what I'm going to do. Even if I do this, if I do it with my cerebral cortex, it's voluntary and conscious. But if, for instance, I see something coming toward my head, my visual reflex will be to move away from it, right? That doesn't involve my cerebral cortex, and because it doesn't, it's actually way faster. It goes straight in, straight out, right? And it doesn't involve very much processing. You don't think about it, and your reflexes are way faster than your voluntary motor activities. So what's an auditory reflex? Same kind of thing. Boom, move, right? Okay, so they are not conscious. They are old school protective sort of reflexes. Did you have something? No? Okay. So that's what your superior and inferior colliculi do. They're visual and auditory reflexes. Does it make sense to you that that's an older functionality than voluntary motor control? Yeah, it's older functionality. And then the cerebral peduncles, right there or right here, those, you can look at them and it kind of looks like they would be mostly white matter tracks. And that's true, they are mostly white matter tracks. So what they are doing is mostly carrying information, motor information, for instance, from the parts of the cerebral cortex that are doing motor information, like those precentral gyrus, right? From there, I've got to get it to my spinal cord if I want to get it out to my hand, right? That makes sense? So that goes through the white matter tracts of the cerebral peduncles. And if the white matter tracts are mostly going cerebral cortex to spinal cord, what kind of tracts are they? Yeah, they're projection tracts, exactly. Does that make sense? Okay, so that's what those are, okay? So your cerebral peduncles. And then for the first time, we're going to start talking about cranial nerves. Now, most of the cranial nerves are going to attach to the brainstem and down, with the exception, of course, of the olfactory nerves, which olfactory nerves just go on the inferior part of the cerebrum way up there. But everything else you're pretty much going to find from here on back. Right? And so we'll talk about cranial nerves as we go through. You are going to have to kind of learn the cranial nerves on your own because it's one of those things that you just kind of have to memorize. But I'll give you some ways to do it. First off, with the cranial nerves, I really like to know where they're attached, right? Because that doesn't vary very much, and if you can follow them back to their attachment point, you can sort of confirm that it might be the one that you're talking about. Like, nobody's really going to miss the olfactory nerves, right? But it's also good to know that the oculomotor nerves are just at the top of the pons, right? And where the other ones are attached as well. So your first cranial nerves that you're going to run into are way up here, and then you run into the optic, right? And then you run into these two, which is the oculomotor and the trochlear, which attached like right here, close to the brainstem. Before we go, actually, I'll do that in a minute. When we talk about cranial nerves, I'll talk about that. Okay, so now we are moving back. So the mesencephalon, or the midbrain, includes the superior and inferior colliculi and the cerebral peduncles. And now we're moving down, and we're going to move down to the pons. Okay, so the pons is here. It's super easy to find, and it's super easy to spell, right? So if I ask you a question on the next test, and the answer is pons, I'm being super nice, because I could have asked you mesencephalon, right? But I am asking you pons, so don't put a D in it. P-O-N-S, no D. So let's talk about where the pons is. Mid-sagittal section, it's also really easy to find. You can see it right there. Here, it's easy to find the pons. And when you look at the pons, you can, let's see, where are we? Okay, so do you see that the pons has both gray matter and white matter in it? Can you see that? And you can kind of see it here as well. So what is the pons doing? What's the pons all about? Well, the pons has gray matter and white matter. And you can see really that some of the gray matter tracks are going, white matter tracks are going this way, and some of them are going up and down. So let's talk about the white matter within the pons. So the white matter tracks in the pons allow communication between the cerebellar hemispheres. So when the cerebellum communicates from one half to the other, although it seems kind of counterintuitive, it actually sends the tracks through the pons. So what kind of tract would that be? You can do it. It's not projection, it's not association, it's commissural tracts, yeah. So that's a commissural tract that goes that way. But then it also has white matter that connects the cerebrum and the cerebellum, right? So that would be a projection tract, right? So that's what you're kind of seeing in this picture, right? It's kind of suggesting, although don't worry, I wouldn't have you look at the figure and tell me the tract from looking at the figure, but you can kind of see that you can see the commissural tracts and projection tracts running through the pons. And then the gray matter nuclei within the pons, remember if you've got a wad of gray matter within the central nervous system, it's called a nucleus. Okay, now I will not ask you to individually identify them. The reason I'm showing you is these nuclei, their names kind of tell you what they're doing just in case you run into them in your future. Do you know what the prefix PNEU means? Air, exactly. So are you at all surprised that the pneumotaxic and the apneustic center are involved with respiration? Right? Does that make sense? So we have moved down to the point that we are talking about life-sustaining functionality here, all the way down in the pons of the brainstem. Does that make sense? So you've got the pneumotaxic and the apneustic. I will not ask you those, just so you know. I just want you to know that the gray matter in the pons is related to regulating respiration. Okay, so that's the pons functionality, nuclei that control respiration. You've of course got more nerves, cranial nerves attached there. And then we are going to move down to the medulla oblongata.
So 5,
Go ahead.
6, and 7 are generally with respiration?
No, they're not related just to respiration. They're related to a bunch of different things. They just happen to be coming and going from there, not particularly relevant to respiration. But in physiology, for instance, when you start talking about control of respiration, you're going to start talking about the pons. Like, okay, how do I know when I need to increase or decrease respiration? The whole feedback loop goes through your pons. Likewise, when we're about to talk about the medulla oblongata helps with respiration, but also does everything cardiovascular. So the good anatomy student can carry that information into physiology, and when you start talking about regulating, for instance, blood pressure, how do you know when to increase or decrease your blood pressure? It goes through your medulla oblongata. That's what it does. So that's why this stuff like ceases, hopefully if we do it correctly, if I do it correctly, it doesn't stay isolated. It ends up going with you to be relevant to other things. So the medulla oblongata. So here is your medulla oblongata. It's just below your pons, everything below your pons, but above your foramen magnum is considered medulla oblongata, and it's thicker and then it gets thinner. So not surprisingly, the medulla oblongata is also gray matter and white matter. Yeah? So it's going to have some white matter because you got to like connect the spinal cord to the brain somehow. So what kind of tracts do you think are most common in the medulla oblongata? Yeah, it's mostly projection tracts. The white matter is mostly projection tracts. And then the gray matter and the medulla oblongata does a couple of different things. The two most important things for you guys to know is that it also helps with respiration, and then it controls the entire cardiovascular function. Okay? So it ends up becoming very, very important physiologically. So now what we have done is just told the story of everything from uniquely human personality and intellectual functions all the way down to life-sustaining functions like rage and reflex and sex drive, and all the way down to really life-sustaining functions like respiration and cardiac function, right? And then from there on, we're just going to move down to the spinal cord in the next set of notes. Does that make sense? Okay, so as far as locations of things, I think that the ventricles are nice and helpful for finding locations. If you are, like I said, if you are in the cerebral or mesencephalic, do you see why it's called the mesencephalic aqueduct now? because the region that's just around it is actually not the cerebrum. It's the mesencephalon, isn't it? Right here, right? That's why they want to switch the name. The problem is that I teach you the aqueduct before I teach you the mesencephalon, so it just seems arbitrary and kind of torturing. So mesencephalic aqueduct is actually a better name. So here's the mesencephalic aqueduct. When you're around there, you're in the mesencephalon of the brainstem, right? And then when you move down, the pons and the cerebellum are around the fourth ventricle, right? And then you're going to continue to move down, and the central canal is down here. Even though you're not in the spinal cord, it's still called the central canal down there. Okay, so the other cranial nerves, the rest of them actually attach near the medulla oblongata. And those are the hard ones to me. These guys right down here. I will tell you that, I mean, you know I'm going to ask them off of a figure because I'm not going to have wet specimens in lecture. But you're going to need to ask your lab instructor how they would ask those nerves. Because if they're going to ask you off a wet specimen, you may need to have them help you figure out how to tell them one from the other, right? And there are some lab instructors that never use models. So if they're going to ask them off, I would need some help. So how do you know when you're in a cranial nerve number 11 versus 12? How do you tell? And they should have a dependable way to show you how to tell if they're going to ask them. But when I taught lab, and I don't teach lab very often, but when I taught lab, I always asked these off of wet specimens and these off of models because I don't like to be a big beastly bastard. The fourth ventricle is between the brainstem and the cerebellum, and then you can do all of that labeling. Okay, so now the last thing we're going to talk about is the cerebellum. And the cerebellum is kind of posterior. And interestingly, it's actually not the oldest thing. It kind of develops almost sort of consistently with the cerebrum, but like lateral to the cerebrum. It's kind of developed sort of at the same time. It is older than the newest cerebral functions, but it's not as old as the oldest brainstem functions. So, of course, when you look at the cerebellum, you can see that it has this gray matter, white matter arrangement, gray matter on the outside and white matter on the inside. And notice that the folding of the cerebellum is totally different than the folding of the cerebrum, right? And the cerebrum, they're called gyri and sulci. Here they're called folia, and folia means leaves because they thought it looked kind of like an evergreen tree. It's got like those sort of like evergreen branches that you have there. Does that kind of make sense? You see? So those are called folia. And then if you look at the structure here, there are actually two cerebellar hemispheres, just like there are two cerebral hemispheres. And the vermis is this little worm-like structure that goes between them. So you've got the right cerebellar and the left cerebellar hemisphere, and then the vermis that goes between them. And then the gray matter, I'll talk about what it does in just a second, and then the white matter. The white matter on the inside forms that like tree-like structure. And so somebody named it the arbor vitae of the cerebellum, which means tree of life. Because when they named it, they actually thought it was life-sustaining functions back there, because everything else down there is life-sustaining functions. But it's actually not, so we'll talk about its functionality. So let's see if we got everything. Folia, cerebellar hemispheres, yeah. Okay, so the function of the cerebellum is kind of interesting, because a lot of functionality that the cerebellum does, people think happens elsewhere. The cerebellum is actually smoothing and coordinating motor movements and maintaining posture and equilibrium. What do I mean by smoothing and coordinating motor movements? You know what people always refer to as your muscle memory? It doesn't occur in your muscles, it's in your cerebellum. So here's what happens. When you first learn how to do some kind of physical activity that involves some kind of pattern, right? Like learning to play tennis. Every movement that you do when you're initially learning involves your cerebral cortex making conscious voluntary decisions, right? Because you've never done it before. You're like, I don't know how to hold this thing. Where do I put my hand? What do I do? Where do I put my feet? What do I do? So that's all cerebral cortex activity. But after you have learned it very, very well, the pattern is relatively dependable. It's got minor variations, but you've even calculated in the minor variations. And then what happens is after you've learned the pattern very well, you can initiate it with the cerebral cortex, and then you can pass it off to your cerebellum. And that frees up the super valuable processing space in the cerebral cortex and passes it off to the cerebellum. And the cerebellum continues the pattern, and it actually does it more smoothly than doing it voluntarily. Anybody who's ever done any athletic activity knows that once you know it, you try not to involve your cerebral cortex in doing it very much. Otherwise, it makes it really clunky. Like you get in your head too much and then you suck, right? So you have to just like get it started and then enough practicing that you pass it off to your cerebellum. So it remembers these patterns. So let's think about it when you're like starting to ride a bike. So you got a 5-year-old and you're teaching them to ride their bike. And everything is kind of clunky and they're like involving their brain and they're falling over and they're trying to figure out like when you put this foot up, when you put that foot down. But then after they learn to ride their bike, like you riding your bike, you just get on the damn thing and you just go. And you could think about 3,000 other things while you were riding your bike until a cat runs in front of you. And if the cat runs in front of you, what happens is your cerebral cortex clicks back in, adjusts the situation, gets you out of the way of the cat. And as soon as the pattern is familiar again, your cerebellum clicks back in again. And you can see how passing that off really like frees up this valuable real estate. And this one actually does it better than your cerebral cortex. So you run into this in situations that are not athletic as well. If you're doing a repetitive behavior that you've done hundreds of times, a lot of times you really disengage your cerebral cortex. It's not completely disengaged. It's actually taking in information. But when you drive home and it's not snowy and you're driving home the same way that you have driven home 7,000 times before, and you get there and you have no recollection and you were not drunk, right? You're like, I have no recollection of driving. So what happens is you kind of got some anecdotal evidence that you really disengaged your cerebral cortex in the entire process, right? You just like clicked it in whenever you like had to pay attention to a red light and then clicked it right back out again, and then clicked it in whenever you like somebody slowed down in front of you and then clicked it right back out again. So your cerebellum was doing most of that sort of pattern the entire time. That's where your muscle memory is. I have one little story that that sort of taught me when I was first teaching this what the cerebellum does, because the first one of the first semesters that I taught in Texas, I was a brand new faculty member, and I had a student. She was like 5 feet tall. She's a little bitty tiny thing, and she was in my anatomy class. And she had been a cheerleader in high school, and because she was so tiny, she was usually the one who was on top of the pyramids, right? Just like way up there on top of the pyramid. Well, she fell off one too many times, and the last time she fell off, she actually hit her cerebellum, and she actually had a concussion on her cerebellum. And how did that manifest? Well, it manifested in the fact that she lost all coordination. She was no longer a cheerleader, and for that matter, she actually had to reteach herself how to walk. And I learned this because she was trying to walk with me from one class, and she wanted to ask me a question, but I had to go to another class. And so I said, can you walk with me and we'll talk about this? And she's like, no, I actually can't, because she literally, and I mean literally, could not walk and talk at the same time. She would have to stop, because then we talked about it later, and I was like, so what's going on in your head when you're walking? And she's like, well, I don't say the words, but I am actually thinking left foot, right foot, left foot, right foot, left foot, right foot, because she like no longer could pass it off to her cerebellum. So everything was involving cerebral cortex activity. And so it was 2 years later. They expected her to regain functionality, but she hadn't regained it all yet. So she was still really young. She was like 19 years old. So hopefully she would have gotten it back. I don't know her anymore. But so anyway, that's what your cerebellum does for you. It also does these important like minor postural corrections and equilibrium corrections to keep you like sort of like this subconscious motor activity is what it does. So anyway, that's the cerebellum. Okay, and then the last thing is the cranial nerves. So let me just introduce you to how I want you to study the cranial nerves, and then you're really going to have to do this on your own. There are 12 pairs of cranial nerves. I will not nitpick you on left and right because this is hard enough. Because what I want you to do with the cranial nerves is I want you to know the Roman numerals and the names of each one of them. And sometimes the names are helpful, right? Like this is the olfactory nerve. I'm pretty sure you know what that does, right? Okay, yeah, it does smell. And sometimes they're not helpful, like abducens, that doesn't really help you with anything, or trochlear. So know the numbers 1 through 12 and know the names. And then you have to put together some simple functionality with it. How simple? Well, I wrote the lab list, and that's what I want you to know, right? It has simplified motor activity and simplified sensory activity if there is any. So as far as all these nerves go, some are sensory, some are motor, and most are mixed in functionality, right? So if they're mixed in functionality, you need to know the sensory and the motor, right? Okay, but it's simplified. I made it as simple as I possibly could. And then the other thing is that you need to know the whole that it goes through. And the reason I'm torturing you with this is because way back at the beginning of the semester, when you learned all those freaking holes in the skull, and you had no idea the significance of the holes, I am missing the opportunity if I do not have you put back together the holes with the things that go through the holes, right? So I want you to go back and put that back together. And it's all in that same table too, right? I put it all in one table for you. It's got the foramina that it goes through. And sometimes it's like really obvious. The optic nerve does vision, right? Goes through the optic foramen, and it is cranial nerve number 2, right? But sometimes it gets a little harder. Sometimes the name is implied or the function is implied, but it's not told, like the vestibulocochlear nerve. Yeah, it does stuff related to the ears. This week, when you do, if you didn't do it on Friday, when you do the eye and the ear, you'll learn the vestibule and the cochlea of the ear, and it won't be very hard for you to put together that does hearing and equilibrium. Go ahead.
If you have the name, what's the purpose of the number? I'm curious, do people use the numbers?
Yes, people use the number all the time. Yeah, they use them in physiology, and they actually use them clinically as well. So the numbers are just as likely to be used as the name. And I can't tell you what you're going to run into in the future, so I don't want you to like not have the information that you need. So that's what you're going to need to know about the cranial nerves. But I will promise you this, that if I ask you cranial nerves on a diagram, that it will look very much like this diagram, right? Which I acknowledge is not true anatomy, but it's hard enough, so I'm going to make it relatively familiar to you guys, right? So you have to learn basically the same things in lecture and lab. Okay, that good? All right, so something else I was going to tell you. Oh, also, I don't know if you guys looked last week, but since you're going back this week, and you're going to be looking at the spinal cord this week, I want you to know that go back and look at the brain, because we have some really cool dissections of the brain if you didn't get to see them on the cadaver. You guys all got to see human brains, right? There are human brains on both campus. If you didn't get to see them, then ask this time and make sure you do get to see them, because we have real human brains on both campuses. In addition, we also have some cadavers, at least one on each campus, that you can remove the brain, which is a cool dissection. And the reason it's a cool dissection is because it does what I wanted you to do. It puts together the skeletal and the nervous system, because this is how we did the dissection, and it was my lab aides and I that did this dissection. It was really fun. So you get a bone saw and you cut off the top of the head, and then you got the top of the head, and then you flap the dura back or remove the dura, whichever you want to do. And then you pull the brain up. And then it took like 3 of us, because somebody had to pull the brain up and somebody else had to be down there clipping the cranial nerves and pulling it off the cristagalli of the ethmoid bone. But the cool part is, if you did it well, and it's really good on at least one in each campus, you can see the cranial nerves here, and then you can put them right back into the holes, right? And you can pull it up and you can put it right back into the holes. So it really like reinforces that skeletal system and cranial nerve connection, which is what I would like for you guys to be able to reinforce. Does that make sense? Okay, so am I going to ask 20 questions about this? No, but there will probably be 4 or 5 on the next set of notes, because I want you to do that, okay? Okay, maybe 3, 4, or 5. All right, so that's that. And then the next set of notes is what you're going to be doing in lab this week. Well, part of what you're going to be doing in lab this week. So in lab this week, you are doing spinal cord and spinal nerves. And you're also doing eye and ear. I don't do eye and ear in lecture, so the only time you'll get those is in lab, but I do spinal cord and spinal nerves. It's just because you run out of time in lecture, and I need the time for cardiovascular system. And a lot of you guys are not going to end up working with the eye and the ear, but I know that it's very likely that cardiovascular system is going to be relevant for a lot of your careers. So I had to choose. All right, so spinal cord and spinal nerves, which is chapter 16, and then the autonomic nervous system, which is chapter 18. So we'll do chapter 16 today. So the spinal cord. Let's look at what we're talking about. So from here, right, if we start moving down, when does it become spinal cord? Arbitrarily, it really becomes spinal cord after you pass through the foramen magnum. But does the top of the spinal cord look exactly like the bottom of the medulla oblongata? Yes, it does. So you're not going to run into anything that looks dramatically different than the bottom of the medulla oblongata. It's just considered spinal cord after that. So you are spinal cord once you leave the foramen magnum, and then the spinal cord actually ends way higher than people expect it to end, because the whole vertebral column goes all the way down to the sacrum, and it's got the hole in it all the way down to the sacrum. The coccyx doesn't really have a hole in it, but so a lot of people think that because the vertebral column is that long, then the spinal cord is that long, but it's not. Spinal cord ends about L1 or L2, ends right about here. And so it ends up being about 16 to 18 inches long on average, but that's just average, because if you're as tall as he is, it doesn't just end right up here. It goes till L1 or L2, right? So it's like on average. So, and the really interesting thing about the spinal cord that I was so surprised by is how tiny it is. It's like half an inch wide. And it seems like, you know, like half an inch wide. It seems like for that important functionality, I expected it to be like that big and have red warning signs on it. Do not touch, do not touch. But no, it's half an inch wide. It's completely anatomically unimpressive. So go look at it. We did this dissection. There is a dissection kind of like this on Redwood, but there's one just like this on Jordan. And the reason there isn't one just like this on Redwood is because it was like incredibly hard to do. So it took 5 of my lab aides about 6 months to get this dissection done. And what they did is they took off a little bit of the occipital bone so you can see how the brain led into the spinal cord. And then for every one of the vertebra, they took off the spinous process, right? They took off the entire arch and the spinous process. And then the problem is that you can't really use a bone saw because you can't tell depth. And so you have to like really use hand tools to get every single one of them off. And then once you get down here to the sacrum is really thick. It's hard to get through it. And again, you can't use any automatic tools because you're going to cut through way more than you intended to. So like carefully they had to like piece every little piece off as they move all the way down. So that dissection is done, and you can see all of the nerves coming out of it. It's on Jordan campus, and there's one similar to it, but not quite as detailed on Redwood campus. So it's half an inch wide, it's about 16 to 18 inches long, and ends about L1 or L2. And when you look at the spinal cord, just to show you this, we'll look at it in a lot of detail today. If you did a cross-section of the spinal cord, it's generally white matter on the outside and gray matter on the inside. So it's kind of opposite of the brain. The brain has superficial gray matter, and the spinal cord has superficial white matter. Okay, so if you look at the entire spinal cord, the other thing that's clinically significant is that even though the spinal cord ends at L1 or L2, the meninges do not end until about S2. That means that the meningeal sac is still going, and there is nervous tissue below where the spinal cord ends. It's just that it's PNS tissue and not CNS tissue. It's actually nerves and not spinal cord. Okay, so this becomes clinically significant because remember when we were talking about epidural injections? Now you can see why they almost never do epidural injections above L2, right? Because you don't want to accidentally damage central nervous system tissue. And now you can see why. So let's say I thought that I had some kind of pathogen or some kind of problem with my central nervous system tissue, and I wanted to look at the cerebrospinal fluid before I did any biopsy, right? Like, because the cerebrospinal fluid is relatively non-invasive, at least comparatively speaking. So where do I get cerebrospinal fluid? Well, in theory, just theoretically, I could go through my cranial bones and get it from the subarachnoid space, but hell no, right? I could go all the way into a ventricle and get it, but hell no, okay? Or I could go into the subarachnoid space, not even around the central nervous system tissue. And remember, it's a circulating fluid, so it can give me indications of what's going on in my entire central nervous system tissue. So when they go down there and they suck up cerebrospinal fluid, it's called a lumbar puncture or a spinal tap. Lumbar because that's usually where they go in. And what they're going to do is insert a needle between L3 and L4 or L4 and L5. Go ahead.
I'm confused why you're doing that because the spinal cord is not there, right?
Yeah, you don't want to risk damage because you have to go all the way into the subarachnoid space, which is just superficial to the pia mater and the actual spinal cord itself. So it's even more dangerous than an epidural because you're actually puncturing the dura mater and going into the subarachnoid space. So if you went, a couple of centimeters too deep, you would end up damaging central nervous system tissue if you were around the spinal cord. Does that make sense? Now, it's not impossible that you could damage nervous tissue, but it would be PNS tissue if you damaged it down here. So between L3 and L4 or L4 and L5, and usually they get a good spread between your vertebrae by doing this, and you have to go through which meninges? Dura mater and into the arachnoid, right? Into the subarachnoid space. And then they suck up their cerebrospinal fluid. So what can they tell us from cerebrospinal fluid? Well, from cerebrospinal fluid, you can examine the chemical content because we know what the chemical content of the cerebrospinal fluid should be, right? And assess whether the chemical content is off, and maybe that's what's causing your central nervous system symptoms. You can examine it for microbes, bacteria, viruses. You can examine it for white blood cells that indicate that there is some kind of infection, and we'll find out that white blood cells, a certain kind increase with exposure to bacteria, a different kind with exposure to viruses, a different kind with exposure to multicellular pathogens. So white blood cell content can give you some inclination as to what's being on. Sometimes also cancer and precancerous cells will mobilize into the cerebrospinal fluid, because one of the interesting things about cancer is that it doesn't stay put. And when it actually breaks loose on its way from point A to point B, it will go into the cerebrospinal fluid. So you can use this as an initial indicator of what might be going on with the central nervous system tissue without being invasive. Now, it hurts like hell from what I've heard. I've never had it done. Has anybody had it done? It's usually, I didn't have it. Anybody this entire semester has had it done. Go ahead. Wow. For what types of what types of cancer do they do chemo in the CSF? Wow. They put chemo in the CSF. Oh my gosh, that sounds terrible. So the things that I do know about spinal taps is that it's a big beastly needle. It's a long freaking needle, and you have to get some elbow grease in to get through the dura mater. And a lot of times you hear it pop when it goes through the dura mater. And then disturbing the volume of your CSF can give you the worst beastly headaches ever, and like entire body and spinal cord brain soreness. And then if the hole doesn't seal on its own, you have to go in and put a blood patch on it, because if you're leaking cerebrospinal fluid, it's going to continue to have symptoms associated with it. So the most common reason that I have encountered for doing a spinal tap is because they fear meningitis. And meningitis is either a bacterial, usually bacterial or viral infection that causes irritation or inflammation of the meninges. And have you guys heard, there's been a couple schools around here over the last 5 years that have been shut down because of meningitis scares. So meningitis causes an irritation or inflammation of the meninges, and it's really scary for a couple of reasons. The first reason is that if you irritate the meninges, remember they're continuous around the brain and the spinal cord. They can cause paralysis if you end up damaging the spinal cord because of the inflammation. They can cause like paraplegia if they happen down here, or quadriplegia if it happens all the way up here, or it can actually cause brain damage and death if it happens around the brain. So it's really serious. And the other reason that it's really serious is it's highly communicable, especially the bacterial meningitis is highly communicable. So it's really easily carried from one person to the other. Now, so there's also viral meningitis. The bacterial meningitis is highly communicable, but it is treatable with antibiotics because it's bacterial and we know what kind of bacteria it is. Okay, but if a kid has bacterial meningitis in the school, sometimes they shut down the entire school just to make sure that there is not any exposure from one kid to the other. Viral meningitis is less likely to be deadly, but it's also harder to cure because it's a virus, right? And antibiotics don't work on viruses. So with the viral meningitis, you are hospitalized, you are quarantined from other patients, and then you are treated and try to keep the inflammation down until your immune system kicks the virus's butt, because you can't do anything. really, about viruses. You have to wait for your immune system to take them on. So that's meningitis, which is usually the reason that they test for that. I have one little personal story about meningitis that when I was in graduate school, we had graduate picnics in our departments, and there was biochemistry, molecular biology, biology, anthropology. We were all in the same building. And so there's like 60, 70 graduate students and all of our professors, and there's barbecue and beer and frisbee and all that stuff, because that's how college is in other states, just so you know. So you might want to leave. So we were playing frisbee, and there was this guy, and I don't know him super well because he didn't work on my floor, but I did know him, and he was a graduate student with me, and we were playing frisbee, and that was on a Saturday, and then on Monday he didn't show up to his lab, which is a super big deal in graduate school because basically you are someone's slave and you are always on call and always there for your graduate advisor, always. Like there aren't any weekends unless they tell you you get a weekend, and you are there every day without exception. So he didn't show up on Monday. And so the people who worked with him in his lab were like, I don't know what's going on. And then his graduate advisor was looking for him, couldn't find him, and they called, couldn't find him. And so that was Monday, and then on Wednesday, they finally went to his apartment to look for him, and he was unconscious on the couch, and they immediately got an ambulance, and they couldn't wake him up. And when he finally woke up, he had already been diagnosed with meningitis, and he was a paraplegic. And they never figured out where he got it. And 2 people in his lab had to get spinal taps, and the rest of us had to not teach our classes until we knew that we weren't going to manifest any symptoms. So it was super fast and serious, and they never figured out where he got it. So poor guy. He actually came back and finished graduate school though. He was it sucked, but he was still functional. Okay, so that's a spinal tap. Now, coming off, do you see these nerves that are coming off right here? So they're coming off on either side. So nerves that are coming off the spinal cord are called spinal nerves, in the way that nerves that are coming off the brain are called cranial nerves. Does that make sense? And there are 31 pairs of spinal nerves, and luckily they don't have individual names. They're just numbered for where they come off, so it's not as hard as it initially seems. So at the end of this set of notes, we will learn the spinal nerves and then what they innervate. But I already told you sort of the start of this story. Generally, the spinal nerves that come off here innervate here, and the spinal nerves that come off here innervate here, and then the ones that do your legs are coming off of the bottom, right? So that's sort of what we'll learn. And then this week in lab, you are going to learn some branches off of each of the spinal nerves, right? You're going to learn some nerves that go to the hands and some nerves that go to the legs and that kind of stuff. So that's what you're going to do. We won't quite get to spinal nerves today, but we'll get to it on Thursday. All right, so let's look at the anatomy of the spinal cord before we do spinal nerves. All right, so when you were looking at the spinal cord and you're looking at the whole thing, 16 to 18 inches long, although this figure doesn't adequately represent it, what I want you to know is that this cervical region of the spinal cord is slightly wider than the thoracic region of the spinal cord, and then it gets slightly wider again in the lumbar region. So this region would be called the cervical enlargement, but to me it doesn't look any larger in this picture, right? It doesn't actually look larger, but it's just slightly wider. And then the thoracic region is relatively skinny, and then this little region right down here is slightly wider as well. So cervical enlargement and lumbar enlargement. Okay. Anybody want to hypothesize why it might be wider in the cervical region and in the lumbar region?
That's where your arms and legs.
Yeah, so you've got all of these fibers going to and from your arms and legs up here. And then in the thoracic region, think about what you're innervating from the thoracic region. There just isn't a whole hell of a lot, because remember that the vagus nerve, well, actually I didn't talk about this, but let me tell you about the vagus nerve. Back up a little bit. So this nerve right here is one of the most physiologically important nerves. It's cranial nerve number 10, which is the vagus nerve. And vagus means wander, like a vagrant. So what the vagus nerve does is it, instead of the rule in which cranial nerves do head stuff, this one breaks the rule and wanders down and innervates the vast majority of the ventral body cavity, including the heart. That's why it ends up being really physiologically important. Okay, so the vagus nerve does that. So that means that I just innervated almost all of this stuff, right? And then I'm going back down to the spinal nerves. And what do the spinal nerves have left to innervate? right? They have like intercostal muscles and like sensation from here, but how much sensation do you actually have on this part of your body? Not that much, right? So there's just not as much going on from the thoracic region as there is from the cervical region and the lumbar region. So that's why you've got a cervical and a lumbar enlargement. Lots of stuff going to and from the cervical and lumbar regions. Okay, so you've got your cervical and lumbar enlargement, and then you've got, okay, at the bottom here, and I'm going to show you in an actual cadaver in just a second, but at the bottom here, where you actually run into the bottom of the distal end of the spinal cord, it's called the conus medullaris, which literally means cone in the middle, right? So the conus medullaris, that's the bottom part of the spinal cord proper. So let me show you what it actually looks like. So look here. So here's the spinal cord, and the reason you can tell that you're still in the spinal cord is because the pia mater has all of those blood vessels on it. So that's still spinal cord. And then when you hit the bottom of the spinal cord right here, it's called the conus medullaris, okay? The very bottom of the spinal cord, which is usually about L1 or L2. Now, there is still a projection that's coming off of the conus medullaris. Do you see how it looks like it's almost continuing down with that texture right there? That's actually connective tissue. And what it's going to do is that connective tissue is called the filum terminale, and the filum terminale goes all the way from the conus medullaris all the way down and attaches the spinal cord to the coccyx. So it's stabilizing the spinal cord, but from the bottom of the conus medullaris all the way until it attaches to the coccyx, it's not nervous tissue, it's connective tissue. Okay, so that's called the filum terminale. Filum terminale means last hair. That's the last hair, the last little part. Okay, and the last little part that you're going to see down here is, do you see that even after the spinal cord ends, there's a whole bunch of like white matter coming off of either side of it? Do you see that? Okay, that is a whole bunch of nerves, but it's not CNS, it's PNS, right? Okay, and collectively there's so many of them that are running through the vertebral canal that somebody thought it looked like a horse's tail, so they named it the cauda equina, which means horse's tail. Okay, so it's all that PNS tissue that's running both lateral to and inferior to the spinal cord proper. Okay, and so if you look back here, so conus medullaris is the end, and then the cauda equina, and then the green thing that they're trying to represent going all the way down to the bottom, that is the filum terminale. Okay, and you should be able to see all of those on a cadaver. Okay, on the cadaver that's dissected well. Good? Okay, so that's sort of the gross anatomy of the spinal cord. And then what we need to do is to look at the spinal cord structures and cross-section. This is not hard as long as you remember that whenever you're looking at spinal cord structures and cross-section, the first thing that you have to do is to figure out what's anterior and what's posterior, which is not hard if you have the vertebra. But what if you don't? So you have to come up with a way that's really dependable for figuring out anterior and posterior. And there's a few ways to do it. The first way, your textbook made it impossible to do, but the first way is actually that the anterior, this little depression, is deeper, but they didn't draw it any deeper, right? So they kind of screwed up here because this is actually called the anterior median fissure because it's really deep. And the back one is called the posterior median sulcus because it's really shallow. But they drew them the same way, so it doesn't really help. So what else can you use? There's a couple other things that you can use. Do you see that the gray matter on the inside of the spinal cord has sort of a characteristic shape? To me, it looks like a swallowtail butterfly with the rounded top wings and then the pointy bottom wings. So the pointy bottom wings go toward the back. Okay? They go posterior. Do you see that? Okay, so you can use that, or if you don't like that, or also you have to acknowledge that the shape of the gray matter changes just slightly as you go from the cervical to the thoracic to the lumbar region. It's not dramatic, but it does change just slightly. But there's one other way that's really dependable. Do you see this little lump right here? Right, that's called the dorsal root ganglion. Okay, and this is the dorsal root. We'll learn about it in just a second, but it's always on the dorsal side, so it's always going to be posterior, and you never have one on the ventral or anterior side. So with that in mind, I'm going to show you a few different pictures of the spinal cord. I'm going to ask if you can figure out anterior from posterior in each one. So this one's pretty obvious. Of course, if you have the vertebra around, then the spinous process is always posterior, and the body's always anterior, so that's cool, right? So this one should be easy, right? Spinous process of the body. So this one, can you on this figure tell anterior from posterior, right? So what am I here? That's posterior, exactly. Okay, so a few more. How about this one? So that's posterior or dorsal, and this one, still posterior or dorsal, and still posterior or dorsal. But what about this one?
Well, it's the other way around.
Yeah, it's the other way around, exactly. Okay, so you have a bunch of models of cross sections of the spinal cord in lab, and not all of them have the vertebra around them. So I want you to be able to be really confident at figuring out when you're looking at anterior and posterior. After you can do that, it's all relatively easy. Okay, so let me show you a couple things about the functionality of the spinal cord, and then we'll go back to the individual structures. So if you look at the spinal cord, it does a lot of things, but please note that it's gray matter and white matter, right? Now, not surprisingly, most of the white matter tracks in the spinal cord are mostly projection tracks, going up and down, right? That makes sense? So that's what those would be doing, the white matter. So mostly projection tracks, not exclusively, but mostly. Okay, so what about the gray matter in the spinal cord? Does the spinal cord do any decision making? Yes, it does, but it's not conscious decision making. It's not voluntary decision making, but it does do some things for you. And so let's talk about a really simple loop through the spinal cord. A really simple loop through the spinal cord is called a spinal reflex. Okay, so reflexes, remember, are more simple than voluntary activities, right? So I want to teach you one spinal reflex, and then I'll show you this one. So a spinal reflex is something in which the sensory information goes through and the motor information comes out the spinal cord without involving your brain in the decision-making process, okay? So this is a classic spinal reflex right here. So this is called the patellar reflex, and if I can do it on myself. Okay, so that's the patellar reflex. It's really fast, and it's kind of a one-trick pony, right? It just goes in, goes back out. You always sort of respond the same way. Not a whole lot of intellectual processing, zero intellectual processing going on. So I want to put that together with the anatomy of the spinal cord. So a spinal reflex, this one is poking you in the skin and having you move when you poked in the skin, get poked in the skin. But what's going to happen is, look how it goes through. The sensory information is going to go through, and the sensory neuron cell body is actually in that dorsal root ganglion that we just talked about, right? Because that's what should be in a ganglion, right? Neuron cell bodies. And then what's going to happen is it's going to take this sensory information through the dorsal side of the spinal cord. So it goes in the dorsal side of the spinal cord. And then this neuron right here in the middle can be there. It doesn't have to be there. It's called an interneuron. And the interneuron is in the gray matter, and it should be because it's a neuron cell body. What the interneuron is, think of it this way. The whole complex level of processing that goes on here is a 1 or a 0. Do I respond or do I not? And that's what the interneuron would do. It would say either respond to that sensory with the only motor activity that you can cause or don't, right? But just so you know, sometimes there isn't even an interneuron in there. If you always respond to the sensory with the same motor, you don't really need a switch in the middle, right? Does that make sense? So sometimes the interneuron isn't there, and sometimes it is, depending on how simple it is. And then the neuron cell body for the motor neuron, right, that's going to carry information out to the muscle, that's in the anterior gray matter, right? So, and then it's going to go out the other route. So what I'm saying is the general functionality of the spinal cord is sensory in the backside and motor out the front side. Sensory in the backside and motor out the front side. Does that make sense? Okay. And I don't know why it evolved that way, but it does help me remember that in the brain, sensory in the back, motor in the front, right? I don't know if it has anything to do with it. It just like helps me to remember that. So that's the general functionality that you're seeing in there. Is that all right? Okay, so this dorsal root ganglion right here is going to contain those sensory neuron cell bodies. It also has satellite cells in it, because it should, right? And then the gray matter is going to contain the other cell bodies that are involved in the process, right? The gray matter of the spinal cord. Okay, so now, if we start looking at just labeling the gray matter versus the white matter in the spinal cord, it's super easy as long as you can tell what's front and what's back, because it's just labeled this way. So a gray matter on the outside, white matter on the inside. okay? As long as you can look at this and tell anterior from posterior, which you should be able to do pretty dependably now, then labeling the gray matter and the white matter is easy because these are anterior, these are posterior, okay? So this gray matter right here, these are called the anterior funiculi, or use the term that everybody else uses. They call them anterior white columns, because isn't that what they are? They're like white columns going up and down. Your textbook uses the term funiculus, but I've never seen another textbook that does. So you can call them white columns if you want to. Anterior white columns. These are posterior white columns, right? And then these are lateral white columns, right? Anterior, posterior, lateral. That's easy, okay? And then as far as the gray matter goes, it's all anterior, posterior, and lateral as well. Oh, wait, one more thing about the white. This little area that is connecting the two halves is called the white commissure. Does that make sense? Because commissural tracts generally go side to side, the white commissure, okay? So one more time with the white matter, anterior white column, lateral white column, posterior white column, white commissure. Okay, go ahead. Yes, exactly, because it is actually white matter. It's like white matter within the central nervous system. So that's a good question. Yeah, it's oligodendrocytes that are myelinating right there. That means if you damage them, the damage generally is permanent, right? Because it's oligodendrocytes don't really help. Now, as far as labeling the gray matter, it's also pretty easy as long as you can tell anterior from posterior. They call them horns. So you have anterior gray horns. I call them gray horns. That's how I learned it, but you don't have to put the gray in there if you don't want to. Posterior gray horns, lateral gray horns. Now the lateral ones are sometimes wider and sometimes narrower. It depends on where you are in the spinal cord, whether the lateral gray horns are really easily identifiable. So do you see that they're a little bigger right here than they were in the other figure? And they're kind of big right there as well. So it depends. The shape of the lateral ones just changes from cervical to thoracic to lumbar regions. But anything that's kind of poking out to the side is considered a lateral gray horn. So you got anterior gray horns, lateral gray horns, posterior gray horns, and then this in the middle is called a gray commissure. Usually something commissural is white matter, but this is connecting gray matter from the two halves, right? So that's the gray commissure that connects the two halves. In the middle of the gray commissure is the central canal of the spinal cord, which contains what? What does the central canal of the spinal cord contain? Yeah, it contains CSF, right? So this is the whole like structure function that you're like putting together right now. Is that okay? Okay. Are we good? Some of you guys look confused. I'm not sure why. Okay, so the gray matter, just to reiterate, the white matter is basically cables, right? It's like wiring, most of it going up and down. So notice, interestingly, that that's actually consistent with what you're seeing here. Do you see that when I'm doing this, I'm not even going through that white matter? So that white matter is doing something different, right? It's probably carrying information to and from maybe my legs, or maybe, no, actually this is up in the arm, maybe to and from something, some lower structure. So like the white matter and the gray matter of the spinal cord are not necessarily doing the same things. Like you got cables running on the outside, but you've got like processing happening in the middle. Go ahead.
Does it make sense? I don't get why, because in the brain, the white matter is on the inside,
Yeah.
the spine, the white matter is on the outside. Why does that happen?
It's embryology of folding. It's how it folds during the embryology, but embryology and development. But I don't know why it has to be that way, but I've seen why it ends up that way. I don't know if there's any particular like significance for it being that way versus the other way. I don't know. Okay, so gray commissure we just did, and then the central canal is continuous, right? All right. So the last thing that we're going to do today is to just sort of put this story together with some parts. So I told you that this was a spinal reflex, right? And so what happens with the spinal reflex is all of the decisions are made at the spinal cord level. Now, does that mean that my brain isn't told about it? Like when you do this, you tell your brain, right? It eventually, like you send up notification to the cerebral cortex so that you're aware that it happened, but it's like asking forgiveness and not permission. So the cerebral cortex is not involved in the decision-making process, but you tell it after the fact. It's like you just took the car and you're a teenager and you don't tell your parents when you take it, and you don't ask for it, but you do tell them when you bring the keys back. You're like, I just took the car and everything was fine. It's fine, don't worry about it. So, I have a 16-year-old, but he's so cautious. I don't think he'll ever steal a car. Okay, so peripheral nervous system function. So spinal reflexes and the reflex arc. So what I'm saying is this is all a little loop that goes straight through the spinal cord, but it's got some parts to the loop and none of them are hard. So this is the last thing we're going to learn today. In order to have this response, you have to have a sensation that initiates it, right? And then you have afferent or sensory information going toward, right? And then you're going to go through the gray matter, right? And then you're going to have motor efferent information going out to the effector, right? So this is really just putting together the terminology you learned in the first set of notes, right? The nervous system notes, together with some actual functionality. So that's all we're going to do here. So spinal reflexes and the reflex arc. You have a sensory receptor, and then you are going to have a sensory neuron. So here is a sensory neuron, also called an afferent neuron. And the sensory neuron anatomically is going to run through this dorsal root, right? Or posterior root, dorsal or posterior root. So the cell body of the sensory neuron is going to be in the blank blank ganglion. What's that called? Dorsal root ganglion, exactly. And then the axon or nerve fiber of the sensory neuron runs through the dorsal root. Okay, so this is the dorsal root and that's the ganglion on the dorsal root. Okay, so the axon or nerve fiber runs through there. And then it's going to synapse in the gray matter of the spinal cord, right? And it's going to synapse with an interneuron, maybe, may or may not be present in the reflex arc, or it can directly synapse with the motor neuron if there isn't an interneuron. But regardless, these two are going to be in the gray matter of the spinal cord, right? And that motor neuron, the motor neuron cell body, is going to be in which gray horn? Usually anterior gray horn of the spinal cord. So basically this is consistent. Once we're getting to motor activities, we're more likely to be in the anterior part of the spinal cord. And then what's going to happen is the axon from that motor neuron is going to run through this ventral root, right? And then it's going to run out to the effector, right? So that's all the same stuff that we learned. So the cell body of the motor neuron is in the anterior or ventral gray horn of the spinal cord. And the axon or nerve fiber runs through the anterior or ventral root of the spinal nerve. And then it goes to the effector organ or tissue. Last thing is that flow of a spinal reflex is through the spinal cord only, and it's very fast, but the information is sent to the cerebral cortex so that you can be aware of what happened. But your cerebral cortex does, just interestingly, have some capacity for overriding spinal reflexes. It can't initiate them, but it can override them. Because if you knew, for instance, that somebody was about to do this, I could actually flex my muscles and try to override the spinal reflex. So it can't start it, but it can kind of impede it, interestingly. Okay, so we will stop there for today. Be careful. Hopefully the roads are good by now.
So you said if we can go through your office and check your answers?
Not today.
Not today?
I mean, you can come look right now. Your average is just your 2 tests.
Oh,
Just a second, let me save this before.